Hello everyone, I Arifa welcomes you all in the video lecture of the topic entitled as Role of Bioinformatics in Metabolic Engineering. These are some of the details regarding the topic associated to its respective subject course and let's move forward in this video lecture. Before moving towards the role of bioinformatic tools, I want to give a quick review of what metabolic engineering is all about. Metabolic engineering is basically the field that involves the approach of modifying the metabolic pathways of an organism to enhance the production of desired compounds or to enable synthesis of novel products. It includes the systematic redesign and optimization of cellular processes to meet specific goals, often used in industrial, pharmaceutical, agricultural, or environmental contexts. As we can see, the present slide represents the workflow of metabolic engineering, which directly refers to the steps involved in this process. The first step is target selection. It defines the compound or product of interest that can be any biofuel, any drug, or any pharmaceutical. It also identifies the metabolic pathways involved in its synthesis. The next step is pathway analysis. It analyzes the existing metabolic pathways using computational tools and identifies the bottlenecks or its inefficiencies which needs to be worked on. The next step is genetic manipulation. It modifies the organism's genome to enhance or redirect metabolic pathways. It involves the techniques that includes gene knockouts, overexpressions, heterologous pathway integration. The next step we can see is strain optimization. It involves the iterative cycles of engineered and tested strains which can be resulted into maximizing the product yield. The last step is validation and scaling. It validates the engineered strain on lab scale, and if it is in progressive manner, it can be further upgraded for a scale up for industrial production. Here we can see the role of bioinformatics, and one of the first role is manipulation of biochemical pathways. It involves the construction of graphs that describes the substrate enzyme product transformation. The another one is stoichiometric analysis of substrate product transformation. Then there is retrosynthesis of the product in compliance with the rules of chemical reactions. The other role that it performs is data retrieval. The database regarding metabolic pathways that can be retrieved from our GEC pathway, Metapsych, HMDB, which can be abbreviated as Human Metabolome Database, Reacto, etc. The other role that it performs is strain optimization. It involves the computational tools for strain design such as OptNock. It suggests the gene knockouts to enhance product yield. The other one is flux balance analysis. It predicts the impact of genetic changes on metabolic fluxes. The another one is CRISPR based knockout analysis. It predicts the target sites for CRISPR editing. One of the example is the optimization of E. coli strains for lactic acid production by knocking out the competing pathways. The other role that bioinformatic tools can perform is data integration and analysis. It involves multi-omic integration that combines genomics, transcriptomics, proteomics, and metabolomics to create a systems-level understanding of metabolism. The tools it includes are Cytoscape for network visualization, OmicsNet, and PathPhysio. For fluxomics, it analyzes the metabolite flow through pathways using techniques like metabolic flux analysis. The software that we can use is Cobra Toolbox and CellNet Analyzer. For example, it can analyze the metabolome of Saccharomyces cerevisiae to optimize the ethanol production. The process of designing or manipulating a biological pathway consists of these 5 steps, that are choosing the database, metabolic network visualization, reduction or compression of the metabolic network, searching for a biological pathway or biochemical process, ranking the search result. Choosing the database involves the software such as KEGG Pathway, Metapsych, Metabolome, which involves the all biochemical pathways regarding the desired molecule or desired product. The next one is metabolic network visualization. The biological pathways that can be visualized as complex networks or graphs in which the vertices and edges most oftenly correspond to the biological molecules and chemical transformations. The most common method used for stimulating the visualizing complex biological pathways include chemical transformation and metabolite graph theory, bipartite and multilayer graphs, hypergraphs, and scatter plot matrix. The bioinformatic tools are that are involved in this metabolic network visualization include Metabolic Tinker and Foglight, which are used for the analysis and prediction. These also visualize the metabolic networks as hypergraphs. The next step is reduction of metabolic network. The criteria for classifying these approaches are analysis of the network structure using the data on stoichiometry and kinetics of biochemical transformation. The another criteria is the metabolic network reduction with properties being retained. The next one is searching for a biological pathway that involves the algorithm that are based on shortest path search, depth first search, informed search, breadth first search, and Monte Carlo method, and the tools involved for this search pathway are Pathway Hunter, Symphony, and GemPath. The last one is the ranking the search results. The most common method for ranking the detected metabolic pathway is based on determining the number of reactions involved in the pathway. This parameter can be transformed into a rank function via the shortest path or the pathway with minimal shared flux. The tools that can be used for ranking the search result are OptStoic and PathTracer. These are some of the databases for the data retrieval of biological pathways. The first one is KEGG Pathway. KEGG Pathway can be abbreviated as Kyoto Encyclopedia of Genes and Genomes. It is a resource integrating 16 databases which are categorized into the clusters such as information about living systems contained in KEGG pathway and KEGG module. For genomic information, it is KEGG orthology, KEGG genes, and KEGG genome. For information about chemical compounds, there is KEGG compound, KEGG glycan, KEGG enzyme, etc. And for information about diseases, there are KEGG disease, KEGG drug, KEGG network, etc. KEGG databases also include analytical tools for data analysis, including comparative analysis of genome maps and computations of the metabolic pathways. The another database is Metapsych. This database contains information about the biological pathways, biochemical transformations, and biological participant molecules involving enzymes and low molecular weight compounds, as well as relevant resources in the literature. Metapsych data can be represented in biopacks and pathway formats. The next one is Reactome. This is web accessible database which visualizes the known biological pathways as graphical maps and offers tools for their analysis, including annotations of molecular components, and facilitates searching for systematizing biological information. The Reactome modules contains cross-links to 100 online resources including NCBI Gene, Ensemble, UniProt databases, CBI database of small molecular entities, PubMed database of literature sources, etc. The database and software are open access. The Reactome data model uses a frame-based knowledge representation and consists of classes that describe reactions or entities. Classes have attributes that hold properties of the represented class instances like names or identifiers. The value types contained in these slots can be primitive. The knowledge base of biomolecular pathways is organized as graph database. This graph database has two main advantages of high performance and simpler ways of performing complex queries. Strain optimization involves these steps that are pathway identification, model-based predictions, enzyme and regulatory design, data-driven refinement, dynamic circuit design, and codon and strain optimization. In pathway identification, one can use KEGG or Biopsych to map pathways for the target compound. In model-based predictions, one can employ Cobra Toolbox or K-base for flux-based analysis and to identify genes for knockout, overexpression, or heterologous introduction. For enzyme and regulatory design, Rosetta or AlphaFold tools can be used to engineer key enzymes. It can also analyze the transcriptional regulation with CellNet Analyzer tool. For data-driven refinement, integration with omics data can be done by Cytoscape or MetaboAnalyst to refine pathway models and validate experimental results. For dynamic circuit design, stimulation of synthetic genetic circuits using iBioSim or Tinkercell to dynamically control metabolic flux. The last one is codon and strain optimization that can be done by heterologous gene expression using Optimizer or Genius. The last one is implement and strain modifications which can be done through the tools such as CRISPR-Cas9 design. Strain optimization can be explained by practical examples such as production of lactic acid in E. coli, which includes pathway analysis by the use of KEGG software, which identifies the glycolytic and lactate production pathways. The next one is flux prediction, which can be done by Cobra Toolbox that predicts the knocking out competing pathways such as ethanol production that wholly increases the lactic acid yield. The next one is enzyme engineering, Rosetta tool optimizes the lactate dehydrogenase for higher catalytic efficiency. The next one is codon optimization. The tool optimizer tailors the lactate dehydrogenase sequence for E. coli codon usage. The last one is validation. This involves the tool metaboanalyst that analyzes the metabolites confirming increased lactic acid levels. The next role that bioinformatic tools perform is data integration and analysis. It involves omics integration and network modeling. Omics integration wholly involves different variant fields such as genomics, transcriptomics, proteomics, and metabolomics in order to provide a holistic review of cellular metabolism. The next one is network modeling. It can be done through tools like metabolic flux analysis and genome scale metabolic models. It is used to stimulate and predict the metabolic behavior, such as predicting the stoichiometric calculations through various methods. This slide represents the kind of data bioinformatic tools deals with regarding the metabolic engineering. The first one is genomic data. It provides the prototype of the organism regarding the identification of enzymes, regulatory elements, and potential modification sites. It involves the tools such as genome sequencing platforms, annotation tools like PROCA, and genome browsers. The next one is transcriptomics data. It captures the gene expression levels under different conditions or after the engineering the strains. It involves the tools such as RNA sequencing for expression profiling, test sequence 2 for differential expression analysis. The next kind is proteomic data. It involves the quantification of protein abundance and post-translational modifications. The tools which are involved in this category is mass spectroscopy, which involves the tools like MaxQuant and Proteome Discoverer for analysis. The next kind of data is metabolomics data. It measures the metabolite concentrations and fluxes in the metabolic network. The tools that can be provided is metabolic analyst for pathway level insights for the characterization techniques such as GCMS, LCMS, and NMR. The last kind of data is fluxomics data. It examines the metabolite flow through pathways. The tools that can be used is isotopomer, which involves the 13 labeled carbon experiments distribution analysis. The workflow of data integration involves various steps, and the first step is data collection. It gathers the multi-omics datasets from engineered and wild-type strains under different conditions, for example, RNA sequencer for transcriptomics and LCMS for metabolomics. LCMS means liquid chromatography mass spectroscopy data for metabolomics. The next step is data pre-processing. It normalizes the datasets to remove biases such as batch effects and sampling inconsistencies. The tools involved in this pre-processing is Lemma for normalization of transcriptomics, Metaboanalyst for metabolomic data pre-processing. For integrative analysis, there is a combination of data from different layers to uncover relationships. The techniques involved for such analysis is correlation analysis and network analysis. In correlation analysis, it links the gene expression with metabolite levels. For network analysis, the tools that are used are cytoscape to visualize the gene metabolite protein interactions. The next step is pathway enrichment analysis. It identifies the pathways significantly affected by engineering. The tools involved are KegMapper and Reactome. The next step is dynamic modeling. It integrates the time series data to model dynamic changes in metabolism. The tools that are involved is CellNet Analyzer for regulatory and metabolic networks. The last step is predictive modeling using integrated data sets to refine genome scale metabolic models and predict outcome for further genetic modifications. The tools that are involved is Cobra Toolbox and K-Base. The final step is validation. This step involves the cross-validation predictions with experimental data to ensure the reliability and the confirmation for using it for the large-scale purpose or industrial purpose. Data integration can be practically explained by the example of production of isobutanol in E. coli, in which the data integration involves the collection of transcriptomics data from RNA sequencer, metabolomics data from GCMS, and proteomics data from LCMS. The analysis involves the correlated gene expression with isobutanol production levels, identifying a bottleneck in pyruvate decarboxylase activity. The next step of model refinement involves the refining of the gem using integrated data, predicting overexpression of pyruvate decarboxylase and acetolactate synthase to improve the yield. The final step involves the validation through the experimental results confirming a 50% increase in isobutanol production. These are some of the challenges faced by bioinformatic tools while working with the metabolic engineering datasets. One of them is biological complexity, which involves non-linear interactions. It consists of the biological systems containing non-linear and dynamic interactions between genes, proteins, and metabolites that are difficult to model accurately. The further challenge is incomplete knowledge. Many metabolites and regulatory networks are poorly characterized, ultimately leading to gaps in the models. The next one is host-specific variability. Metabolic behavior varies between organisms, and tools often struggle to generalize the predictions across species. The next one is predictive accuracy. The simplified assumption involving the tools like flux-based analysis often assume steady-state conditions which may not reflect real biological dynamics. The next one is context-specific behavior. Predictions may fail under specific environmental or industrial conditions due to the unmodeled factors like substrate availability and stress responses. Further challenges that may impose as a drawback includes data limitations, that are incomplete data sets, missing genomic, proteomic, and metabolic data can limit the ability of bioinformatic tools to provide accurate predictions. For example, inadequate annotation of genes in non-model organisms. The next one is data quality. Noise or low quality data from experimental methods such as sequencing errors can propagate errors into computational models. The next one is integration challenges. Different omic datasets have often varying formats, scales, and quality, ultimately complicating their integration. The computational limitations involve scalability. Large scale genome scale metabolic models require significant computational resources, especially when integrating multi-omics data. The next one is dynamic modeling challenges. Most of the tools handle static models, making it difficult to stimulate time-dependent metabolic changes or adaptive responses. The last limitation is limited automation. Many bioinformatic pipelines require manual intervention, which can be time consuming and prone to human error. That's all from my side.